Good afternoon, everyone. Welcome to the Jack Joseph and Morton Mandel School of Applied Social Sciences, and this is the first of our Centennial Research and Training Colloquia series for this academic year. I'm Elizabeth Tracy, Professor and Associate Dean for Research and Training at the Mandel School. This series is co-sponsored by the Office of Research Administration and the Doctoral Program at the Mandel School. And the list of colloquia that I think you all got when you walked in shows the other presentations that we're having this year. We would welcome you to any that you can come to. A few logistics before we get started. Restrooms are to the right in the lobby. Continuing education certificates and the sign-up for professional development hours if you're a social work student are also in the lobby. Some well-deserved thank yous to Ina Brand, who registered you and who did the logistics for this meeting. Maria Sharon, who's in the back of the room, who will be monitoring the participation of those of you who are connecting remotely to this session this afternoon. And Tracy Braden for her help in putting the series together. We will have a question and answer time after our speaker's presentation, and because we are live streamed, we ask you to use the microphone that's up here in the room. If you need us to bring the microphone to you, we will, but we're asking people to come up to the microphone so that everyone can hear your question and the answer. And a warm welcome to the many viewers who are connecting remotely today. You can email questions to mandelschool@case.edu if you have a question during that time period. Today, we are very happy to serve as the host school for the ACES Plus McBride Endowment Distinguished Lecture Series, and that is sponsored by the Deputy Provost Office. I'm glad to say that Dr. Lynn Singer is here today, and also Dean Gilmore from the Mandel School is here today. And the talk will go until 2. We'll have questions and answers before 2, and then from 2 to 3, there's an informal time with whatever cookies are left over that will be in the immediate next room for students who want to interact with the speaker or other faculty. So that's an open time as well. And at this time, let me introduce Dr. Sonia Minnis, who's the chair of our Research and Training Committee at the school and an associate professor, and she will now introduce our speaker of the day, Dr. Gail Richardson.
Thank you, Betsy, and thanks everyone for being here today. I would like you to join me in welcoming our distinguished lecturer, Gail Richardson, Associate Professor of Psychiatry and Epidemiology at the University of Pittsburgh School of Medicine. As Betsy mentioned, her visit here today is supported by the ACES Plus McBride Endowment offered through the Deputy Provost's Office. Gail is a developmental psychologist trained at West Virginia University for her master's and PhD, and the University of Pittsburgh, where she completed a postdoctoral fellowship in alcohol epidemiology. She's worked continuously for over 30 years investigating the effects of maternal substance use, including alcohol, marijuana, and cocaine on the effects of child development and on the development of substance use in adolescents who were prenatally exposed. Dr. Richardson is the principal investigator of the longest-running study of prenatal cocaine exposure. She was just awarded a grant from the National Institute on Drug Abuse to study her cohort now at the age of 27. She has written over 60 peer-reviewed articles on this topic. Through her many years of continuous NIH-funded research and training grants, Dr. Richardson has become an accomplished expert on methodologic issues involved in the study of prenatal substance use, including the assessment of infants, children, as well as adolescents, and the assessment of the numerous environmental factors that can also alter the developmental course. Dr. Richardson was the past president of the Neurobehavioral Teratology Society, now called Developmental Neurotoxicology Society, and serves as an editor for their journal, Neurotoxicology and Teratology. I'd like to remind everyone, again, that there'll be additional time for informal conversation afterwards, as well as a meeting informal meeting tomorrow at the Flora Stone Mather Center for Women about career trajectories for women in academic research. Please welcome Dr. Gail Richardson.
Thank you very much. I'm very honored to be here. Is that okay? I want to thank Lynn and Sonia and Betsy for inviting me and for organizing this. Truly appreciate it. All right, so I'm going to give an overview of 21 plus years of findings. I'm not going to talk about everything. And I want to talk about some of the methodologic issues for those of you that are in training, some things to think about. Those of us that have struggled with it for years are aware of these issues, but it never hurts to be reminded of all the issues. And of course, as Sonia mentioned, I want to thank NIDA, National Institute on Drug Abuse, for funding for all of these years. Okay. So we refer to our study as the Maternal Health Practices and Child Development Project, or the Maternal Health Project is how we refer to it to our subjects. And it's not a formal center, but we run it as a center. There are, I originally went to Pittsburgh to work with Nancy Day. She had two large studies, one of prenatal alcohol exposure and one of prenatal marijuana exposure. And we share the instruments that I use were developed based on what she had used. We share personnel, analytic staff. It makes for a more efficient running of projects. There were, Marie Cornelius also has a study looking at substance use in teenagers. But obviously what I'm going to talk about today is my study on prenatal cocaine use. They are all separately funded protocols. So in terms of what we know about how many women use cocaine during pregnancy, there are very few national data sets that give us prevalence rates of cocaine use during pregnancy. The National Survey on Drug Use and Health reported that 5% of women in this childbearing age group used in the past year, 2% in the past month. The last national data set of drug use during pregnancy specifically was the National Pregnancy and Health Study that was published in '92. That was all ages of women, not just the 18 to 25. And in the national survey, they're showing 1%. Now, these are all separate research projects, so these are not nationally representative samples. But you can see that it ranges from a low of 3% in a very rural sample in Florida to a high of 18% that was an inner city sample. These were the, if you weighted the general screening sample. As you'll see, we selected more than this 8%. But you can see that the rates vary tremendously depending on the locale, and as you'll see, the way that people recruit their samples. Okay. So we all have those of us that do research in this area have our favorite quotes. Early on when we were beginning this study, this was one of the ones that really got under my skin. An article in the School Board Journal, "Blameless youngsters impaired by cocaine before they were born. Won't be easy to talk to, let alone teach." Now, if you notice the date on this, there were no data at all at this time point. And then, as Sonia mentioned, she has this slide in her talks as well. This was a Time article that made a lot of noise about crack kids. And if you look more closely at the pictures inside the article, this child is actually fetal alcohol exposed, okay, if you look at his characteristics. Okay. But that was kind of the context, early on in my work. So there were these dire predictions of what the long-term effects were going to be. So as a good postdoc, I started off, learning about all the issues and delving into the research, and I was shocked at the state of the research early on when I started this. There were a lot of things that needed to be considered. Most of the studies were not well designed. There were a lot of these methodologic problems. And of course, the most serious that was in existence at the time was the lack of a control group. And as we know, case studies are important in the early stages of research for hypothesis generating. But what happened was that the media took these early studies and ran with them in the absence of any data. And that was the crack baby epidemic that received so much attention. So there are many, many challenges in studying prenatal drug use of any kind. Prenatal cocaine use is particularly challenging because it's an illegal drug. But there are other issues that I'll go into some more detail on. But kind of to give you a framework, we use the teratologic model as a basis for our work. We used it as the guiding principle for what kind of exposure information we wanted to get, what kind of outcomes we wanted to look at. One of the first principles is that outcomes are a function of the dose. And in a picture, it looks like this. So these are different domains of outcomes: central nervous system, growth, morphology, mortality. And as the dose increases, you will get more effects in more domains. In addition, as the severity increases of the dose, you will get more serious effects. So this guided our choice of outcomes. And the second principle, that the effects are stage-specific, that it matters when during pregnancy the exposure occurred, guided our choices for assessing the exposure. Okay. You've probably seen this picture as well, looking at critical periods during pregnancy, so that early in pregnancy, an exposure will have an impact on major morphologic abnormalities. And as you get later in pregnancy, central nervous system effects can occur throughout, but also into the third trimester. The third principle of the teratologic model is that effects may emerge later in development. They're also called sleeper effects, and we see some examples of that in our work. And then the last principle was not part of the original teratologic model, but people have come to realize that not only the exposure, but the rearing environment has a tremendous effect as well. Okay. And here's my picture where, Lisa, is that right? Lee, sorry. Okay. So, based on this teratologic model, and when we're planning our outcomes and our analyses, there are several models that you can consider. Okay. The direct model is based strictly on the teratologic principles, that there's a direct effect of the cocaine exposure on your outcome. But as developmental psychologists, social workers, we know that there are many other things that influence development. So we can also consider a moderating model, or an interaction or indirect effects. For example, that home environment might influence this relationship. So only in the presence of detrimental home environments might you see negative effects of cocaine. Okay. And then a mediating model where there are no direct effects of the cocaine on the outcome, but cocaine influences, for example, the mother's current drug use, and that is what impacts the child outcome. Okay? I didn't put in mediating monitoring. We'll leave that till later. Okay. Okay, so this is the design of our study. The women were recruited in the late 80s and early 90s. Now, one of the nice things about living in Pittsburgh, and I won't make Pittsburgh-Cleveland jokes, is that it's conservative, as Cleveland is, I imagine, as well. But the drug use trends are a couple years behind the rest of the country. So that when it came to studying prenatal cocaine exposure, I had the advantage that I could see what was going on in some of the other cities and was able to kind of get right in the beginning of a lot of the cocaine use in Pittsburgh. But I recruited the women from McGee Women's Hospital, which is the large obstetrical hospital in Pittsburgh. And I recruited them from the prenatal clinic. And I did this on purpose because I wanted to have women who had prenatal care. Because we know that prenatal care, women who seek prenatal care are different from women who don't. A lot of the earlier studies recruited women at delivery, and you get a very different kind of sample. Okay? So we recruited them early in pregnancy, and then based on their reported drug use, we enrolled any woman who used at any level, because we didn't have a guideline for what level was going to have a detrimental effect. And then the next woman interviewed who reported no use was also enrolled. Then we interviewed them again. This is at about the end of the first trimester, the end of the second trimester, and then at delivery. And these are all completed follow-up phases. 1, 3, 7, 10, 15, 21, and then, as Sonia mentioned, this is our ongoing phase. Okay. And by the way, I'm happy to, if you've got questions as we go along, happy to be interrupted. The women were 18 years of age and older, English speaking, and they had to have come in for prenatal care no later than their 5th month of pregnancy. So if they showed up for delivery, they were not in this sample. Okay. I do have a subsample of women with no prenatal care. That's a very different group. Okay, there we go. One of the very important parts of a longitudinal study is making sure that you can follow the people. And as you learn the advantages and disadvantages of longitudinal studies, one of the main drawbacks is loss to follow-up. And some of the earlier studies only saw like 30% of people even at their 1-year phase, so it was very poor. So we put a lot of effort into making sure that we do whatever we can for people to participate at each phase. So there were 295 in the birth cohort, and you can see at each of these phases here, this is of the eligible subjects. So babies that died, women that refused to ever participate again were taken out. But at 21 years, we had 90% of the eligible subjects. And if you use the more conservative rates, percent of the birth cohort, at 21, we had 76% of the offspring participating at 21. You can see it's a little higher at 21 than it was at 15. That's because we didn't need the mother's permission anymore. At 15, the kids were still tied to the mothers bringing them in, but at 21, they could bring themselves in, and we were able to have a higher follow-up rate at that point. Obviously, these rates depend on a lot of things. Having a wonderful staff, having good rapport with the women, they get hooked into the study. We do things like newsletters, sending them newsletters 4 times a year, birthday cards, if they call in and ask for help with problems, that kind of thing. And Pittsburgh has a relatively low mobility rate, so the women may move around a lot within the city, but they tend not to move out of the region, which also helps. Okay. We also don't have any differential loss of subjects at any of these phases, the exposed versus non-exposed, no differential dropout. Okay. So, first thing is, how are you going to measure drug use? Okay. It's a critical question. And there's a lot of ways that you can measure drug use. You can interview people. You can have them fill out self-report. You can do biological measures of substance use. And because we were very interested in the teratologic model, we wanted to know when during pregnancy the exposure occurred. So at the time that well, let me do this first. Okay. So there's different parameters for measuring substance use. Quantity, how much they're using, frequency, how often, and then the pattern or the duration of use, how long, which during pregnancy is obviously a critical parameter. So you take something like alcohol, okay, that's legally regulated. We know how much alcohol is in different kinds of drinks. But what's a dose of cocaine? There's the illegal nature, there's variety in types of cocaine being used, powder cocaine, crack cocaine. One of the interesting things that we found is that the terms that you use really matter. When we ask women, we ask them if they use cocaine, we ask them if they use crack. They were very hesitant to acknowledge that they were using crack. They didn't want to use that term. But if you ask them how they're using, then they're really explaining to you that they're using crack. Oh yeah, I put it in a pipe, it's a little rock, and I smoke it. Well, that's using crack. So the more detailed questions you can ask, the more valid answers you're going to get. When I first started this, and different people measure this in different ways. You can measure it by the amount of money spent. You can measure it by frequency. For descriptive purposes, I went to some of the law enforcement and toxicology labs in Pittsburgh at the time and got some conversion measures that we use for descriptive purposes that you'll see in a minute. Okay. So these are our interview questions. Now, the women we don't hand, this is not a self-report, okay? These are administered. But Nancy Day came from the alcohol research field. So she had experience in asking people about their alcohol and marijuana use. So when you approach people and you say, "You don't drink, do you?" Okay, what's your answer? No, of course. And my OB provided a perfect example when I went in for a checkup. He's like, "You're not drinking, are you?" And I started laughing. He's like, "Why are you laughing?" I'm like, "Because you didn't ask that question right." So in all of our substance use questions, we assume that the women are using. Okay? When you use, how much are you using? Okay. So this is the same format that we use for all of the drugs. When you drink, how much do you usually drink? How many drinks, and then how often? Are there days when you have more than 5 drinks? Well, yeah, on the weekend I have 10 drinks. Okay? And are there days when you have fewer drinks than that? Yes, sometimes I only have 1 drink. Okay? Same format, whether it's marijuana, cocaine, and so forth. Okay. Now, in addition to the formatting of the questions, the interviewer matters tremendously. So when we recruited women from the prenatal clinic, we had our own trained interviewers. We didn't go down with our little white lab coats and say, I want to talk to you about your drug use. We selected women that were similar demographically to the participants, and women that were comfortable in talking about drug use during pregnancy in a non-judgmental manner. And they were familiar with the slang terms used for different drugs. So these different arrows here. Okay. So you see that we ask about the usual, maximum, and minimum. And the reason that we did that is because we find that when you ask someone about their usual use, you don't get the full picture. We're only getting not even half of the amount of use when you ask what the usual use is. For marijuana use, we found that asking, are there days when you use more, we got an additional 45% use. And with cocaine, another 50% use. So you're more than doubling the amount of use you get when you give the women an opportunity to answer in the full range. Okay. You can see a different pattern here with cocaine use. Asking if they used less than that didn't add much, so that their usual and maximum captured most of their use. Okay. So this is for the selected sample, the women that we enrolled. And the blue lines are women that did not use. Yellow are women that used less than a line per day. And red is one or more lines per day of cocaine for, as I said, for descriptive purposes to look at the nature of the sample. You can see that the year prior to pregnancy and the first trimester, there were almost 20% of the women were using one or more lines per day, and another 20% occasional users. But you can see that by third trimester, most of the women had stopped or cut back on their use of cocaine. And that pattern was consistent in the postpartum. So that it was, and as I remind you, it was a prenatal care sample. So there were women that sought health care. Most of them were stopping or cutting back on their cocaine use. But of course, one of the issues that we always have to deal with is what's the correspondence between self-report and biological measures. When I first designed the study, at the time it was called a first award. First awards don't exist anymore. But it was a smaller amount of money that I never could have done the study without being part of the larger maternal health practices study with sharing personnel, because I couldn't afford to do it on my own. But in order to get the trimester-specific information that I wanted, I would have had to do urine screens, weekly, and it was not affordable. At the time, hair analysis and meconium were not reliable enough to use, so urine measures would have been my choice at that time. But as I said, it wouldn't have given me the pattern of use that I want. But nonetheless, we always have to talk about the correspondence between self-report and the biological measures. Okay. So we were able to look at misclassification rates in the sample. So I looked at women that had had urine screens conducted by the hospital. And we looked at those that were positive and negative and what they told us on the interview. And what we found out was that all of the women who had a positive urine screen told us that they used. Okay, we had no misclassification in that group. In contrast, the women that were screened but were negative, almost 80% of them told us on our interview that they used. So if you were using urine screens in this kind of a sample to select your study group, you would miss all of these women that told you that they used. So that's my soapbox for, because, yeah, I mean, it's an issue. It varies with the drug, obviously, and it varies with your sample. As I said, this is a sample recruited from a prenatal care clinic. In our sample of women that had no prenatal care, we do have misreporters. There are about 25% that were positive and did not tell us that they used, and the nature of the sample. Okay, so when you're trying to disentangle whether it's cocaine exposure that's having an impact on your outcome, you need to know what else is associated with cocaine use. We also asked the women, as I showed, asked the women about their alcohol use during pregnancy. This time the red bars are 1 or more drinks per day, and the blue are the non-users. And you can see year prior to pregnancy and first trimester, there's only about 30% of the women that were not drinking during first trimester. But by the third trimester, about 60% were not drinking. And the 1 or more drinks per day had really declined. So the same pattern prenatally that we saw with cocaine. But postpartum, you can see that women return to not quite to the same level, but they resume postpartum their drinking. Okay. In terms of the marijuana use during pregnancy, and I'll remind you that this is late 80s, early 90s. Okay, we'll come back to marijuana later, but this was at that time period. Most of the women, almost 70%, were not using marijuana during pregnancy at that time. Again, by third trimester, the use declined, and most of the women did not resume marijuana use postpartum, unlike alcohol. Now, there's another drug that we all know and love, tobacco. Okay. Very different pattern of tobacco use. Okay. The women are not stopping use during pregnancy, and they're in fact increasing postpartum. This is 1 or more packs per day. Okay. One of the most addictive drugs is nicotine, okay, from a public health standpoint. Not many people look at prenatal nicotine use anymore, but very important. Okay. Those slides were for the sample as a whole. Now this is showing for the women that were using, based on their cocaine status, what other drugs are they using. So blue is the women who did not use cocaine during pregnancy. Red is the 1 or more lines per day. And you can see the tremendous difference in the percent that are using tobacco, that are also using alcohol, that are also using marijuana, and using other illicit drugs as well. This is important because we know that each of these drugs has an impact on child development. So you can't look at a group of women who used cocaine and compare them to a group of women who did not use cocaine, no matter what drug. I mean, it would apply to the opiate epidemic now. You can't look at women who used opiates and compare them to women who didn't, because they're using many other things as well. Okay. So we know there's other characteristics that are associated with cocaine use during pregnancy. The women were older. They're less likely to be Caucasian, less likely to be married, had lower family income. They gained, or they weighed less before pregnancy. And even in this sample that was recruited from a prenatal care clinic, they had less adequate prenatal care, the women that used more frequently. And again, all of these factors influenced your outcome. Okay. This was first trimester use, so no use compared to heavier use. This shows women that used first trimester only and stopped compared to women that used throughout pregnancy. And you can see that the women that use throughout pregnancy are even more different than the women that stop. The percent alcohol use in the women that are using throughout pregnancy. Okay, so that's exposure, how to assess exposure. Excuse me. We also had to decide what we were going to measure for our outcomes. So our batteries were selected based on, at later phases, things we had found at earlier phases, things that people in other prenatal drug studies had found, the teratologic model, obviously. And then we were concerned about being consistent across our phases as well. Okay, these are general domains that we look at in the mothers at each of the follow-up phases. Her own substance use currently, obviously demographics, medical history, things that we know that are going to influence her parenting and the child's outcome. Okay, here's some of the offspring domains at each of the phases. And as the offspring get older, you can assess more detail, for example, in neuropsych performance. And as they get older, asking them about their own psychological status and substance use. All right. So in terms of describing the sample, because this was recruited from the McGee Prenatal Clinic, which was an income-based clinic. It's a low-income, low socioeconomic status sample, predominantly high school educated, predominantly single, and evenly distributed between Caucasian and African American mothers, which does not reflect Pittsburgh as a whole, but it reflects the prenatal clinic from which we drew the sample. Okay. The infants, excuse me, at delivery, basically a healthy full-term sample. Okay. As illustration, at the 15-year phase, about 18% were no longer in maternal custody at 15 years. This is not very high. It's much lower than your sample is. Again, different sample characteristics that are related to that. You can see overall, the sample as a whole, the IQ is about a standard deviation below average. Okay. And about 19% of the sample was receiving some kind of special education. Okay. At 21 years, 85% of the sample were high school graduates. This is actually a little higher than the US graduation rate. It is exactly the PA graduation rate, and it's a little bit higher than the Pittsburgh City School graduation rate. About 3% had graduated from college by this age. Okay. Half were working. Most of them still lived with their parents or a relative at 21. Very few were married, and by 21, almost a third of the females already had one child. So I'm not going to go into all of the findings. Time to do that, but this just kind of lays out the different domains across the years. I'm going to illustrate some findings from the growth domain and from the behavior domain. And all of the findings that I talk about are, unless I tell you otherwise, they're controlled, they're regression analyses that control for all of these other things that you need to consider. Okay. These we put in most every model, and then depending on the particular outcome, we add other variables as important. Okay. So in terms of growth, we found differences at birth, as most other studies in the literature find as well, that the exposed children are smaller at birth. About a week, they're born about a week earlier in terms of gestational age. And then to illustrate at 10 years, see there's about a 10-pound difference between the non-exposed kids and those whose mothers were using a line or more per day. And if you look over the years, you can see the yellow lines are the children that were exposed during the first trimester. That you start to get a divergence in their growth pattern over the years, with the exposed offspring growing more slowly. Okay. Illustrate some of our findings in the behavior domain. These are some findings from our 21-year phase. And these are not exposed, or I'm sorry, these are not controlled analyses. These are just descriptive group differences here. But look at the exposed offspring. Half of them have been arrested by 21, compared to 25% of the non-exposed. Okay. 21% have been diagnosed with conduct disorder. These are not just symptoms. This is a psychiatric diagnosis of conduct disorder, so acting out aggressive kind of behaviors. There we go. 68% of them have had sex before they reach 16. Their Wechsler memory scale is lower, and trail making is a test of planning, thank you. Like going from circle to circle here. This is a time measure here. Those were different as well. Okay. So again, if you plot across the years, looking at some of the longitudinal patterns, the CBCL is the Child Behavior Checklist. It's the mother's report of her child's behavior. And the externalizing score looks at aggressive kind of behaviors, acting out kind of behaviors. So you can see at 3 years and 7 years, the children that were exposed had higher rates of behavior problems. And the same at 15, and then increasing here at 21. Okay. Now there's this 10-year-old phase. What's going on here? I'm not sure. We could call it the latent phase with 10-year-olds. That's about the best explanation I have for that. They're not old enough yet. I don't know what's going on there. But you can see that at 15 and 21, the exposed group is continuing to have increased behavior problems. Now, the other thing to notice here is that these scores are not clinically meaningful. The CBCL has a cutoff of, it's about 67, I believe, for borderline clinically meaningful behavior. So we're nowhere near that. But on a group level, these are significantly different. Okay. And then you can start studying the really interesting things. Okay? Substance use as the kids are getting older. So for those of you that have kids that are these different ages, and you start cringing, by 21 years, almost everybody has had some alcohol. That's just, it's not, excuse me, it's more than a sip, but it's any alcohol use. But if you look at a higher level of use, about 35% have had a drink or more per day. In terms of tobacco, 40% of them are using by 21. And this is where I mentioned how marijuana is different now than it used to be when we recruited the mothers. Almost 60% of the 21-year-olds are using marijuana, and 25% are using a joint or more per day. Okay. This is we like to say this is our new alcohol. Marijuana is the new alcohol. Even fewer people are using tobacco in this group anyways. Okay, so it's very prevalent. And we ask the offspring about use in a similar way that we ask the mothers during pregnancy. Okay. So if you look at marijuana use as a function of their exposure status, the yellow line are the offspring that were exposed to cocaine during pregnancy, the red are the non-exposed. And you can see the offspring that were exposed start using earlier, and around about 11, you get this huge increase in the trajectory of marijuana initiation. Okay. For those of you that care, this is a Cox proportional hazards model. And it controls, the other prenatal use, other current use. Okay. Okay. When we start to look at some of the models to try and explain these pathways, we start off with our direct model, and we see that the cocaine exposure is significantly related directly to the offspring's initiation of marijuana use. But we also want to look at some of the pathways of use. So we looked at a lot of different things. We know that exposure to violence is very important in terms of kids' onset of substance use. So we looked at that. We also looked at factors in the home environment, and family history of alcohol problems also predicts the offspring's onset of use. So we found significant indirect pathways, with the cocaine exposure predicting being associated with exposure to violence, and that predicts onset of marijuana use. However, this direct effect, it was reduced, but it's still significant. Okay? So we find both the direct effects and indirect effects on marijuana initiation. We looked at child behavior problems, earlier depression. Those things did not mediate the pathway. It did not mediate this direct relationship. Okay. And then another interesting outcome is what the offspring are doing in terms of risky sexual behaviors, which is going to be obviously an important predictor for detrimental health outcomes later in adulthood. So this is the same format. The yellow line is those that were exposed to cocaine during pregnancy. and the red is the non-exposed. And this is the age at which they reported starting to have sex. This is sexual intercourse. And the percent of the offspring. So again, the exposed offspring are initiating earlier, and they are diverging from the non-exposed as they get older. Okay. And then again, looking at the pathways of use, we found a direct effect between the cocaine exposure and the age of sexual initiation. This was also related to parental supervision. So all of you that want to know where your kids are, keep doing it, because they're less likely to initiate sex earlier. Okay. However, when we started looking at some of the indirect pathways, what we found was behavior problems didn't change the pathway, but the early marijuana use did. So that the cocaine exposure we saw related to early marijuana use, and that is related to earlier sexual initiation. The direct effect was no longer significant. So this was fully mediated by the earlier marijuana use. Okay, so this is a summary of many years of work, just to give you kind of a big picture of findings in the different domains. We do find reduced growth at every phase, even up to 21 years of age. And it's not related to the growth deficits earlier. As I showed you, they continue to grow at a slower rate. Okay. I didn't talk about any of the internalizing kind of behavior outcomes, but when the kids were younger, the mothers rated the exposed infants as more fussy and difficult. That's a common finding in the literature. And as they're getting older, they're not clinically depressed, obviously, but they are reporting more depressive symptoms if they were exposed in utero. And then in terms of the behavior domain, one of the consistent things we're finding is these externalizing, aggressive acting out behaviors, which as you're getting to be an adolescent and a young adult, they are manifested as drug use and delinquency, conduct disorder, sexual behavior that I mentioned. So I talked about what I think are strengths of the study, recruiting from a prenatal care clinic where you control for some of the differences in health-seeking behaviors. We have very good follow-up rates across phases, looking at detailed assessment of exposure, different domains of outcome. We find consistent effects across different phases, which is also an indication of consistency in findings across the different phases. Okay. Most of our findings are related to first trimester exposure. Unlike some other samples that have heavier users throughout pregnancy, the pattern of use in our sample is predominantly first trimester use, and then the women stop. Okay. We don't find a lot of effects with third trimester exposure, but we have very few women that continue to use throughout pregnancy. But this pattern of use is typical of what you see among pregnant women in non-treatment seeking samples. Patterns are consistent with the principles of the teratology model that I showed you in terms of timing. It's also consistent with a lot of other research that's been done in the field. The behavior domain is really the one that seems to be consistent across a lot of studies with the externalizing aggressive kind of behavior. as all of our samples get older, we're now able to look at substance use, and this is becoming a consistent finding as well in the few of us that are still doing this, finding earlier initiation of substance use in adolescents and young adults. And we know that these early behaviors are very important for adjustment to adulthood, leading to poor outcomes in a whole variety of important domains. To pay homage to my animal researcher colleagues, these patterns are consistent with changes in neurotransmitter systems. In fact, we've done one sub-study, and I know a lot of other people are doing sub-studies looking at more direct brain imaging to try and target some of these more detailed systems. And of course, we have to keep following the offspring to look at domains that are going to be developmentally important at each of the different stages. Okay. So this is my new phase. We did many, many happy dances when this phase was finally funded. It was much more difficult, as you can attest to, getting the follow-up funded this last time. But we're no longer assessing the mothers. We're just looking at the offspring at this phase and looking at developmentally appropriate outcomes for this age, adult role development, substance use, psychiatric disorders, and so forth. And we're learning lots of interesting things, both from the staff and from the subject perspective. Drug use is cyclical. It changes. What's in fashion changes. And as I said, we're learning new things to ask about, how to ask about them. A lot of our subjects are vaping now. And one of my colleagues is very interested in this e-cigarette or alternative tobacco use delivery system. These are all different kinds of vaping devices. This is first generation, which looked most like a cigarette. And you can see, I think these are like third generation now. There's these different modules. She's been going to do research at vaping shops. Walks in, I don't want to buy anything, but can you tell me about these different devices? There is, up until a month ago, no federal regulation of these devices. There is going to start to be some, but it'll be a while until the federal regulations are in place. So from shop to shop, the amount of nicotine that's delivered through these devices varies. So talk about an assessment nightmare for how to measure what nicotine people are actually getting. That is a good use of biological assays, is looking at cotinine level. In addition, people are using these devices to vape marijuana. So our 25-year-olds that we're asking about their own drug use, they get them out of their pockets and they say, here, this is my little vial and this is how much nicotine it has in it, and they can tell us about the pattern of use, but as with a regular cigarette, it's going to depend how deeply you inhale and all sorts of factors. And then when it comes to vaping marijuana, in terms of trying to get amount, it's very difficult. And there's all sorts of other kinds of ways that people are using marijuana as well, including marijuana wax. It got its name because of what it looks like. But this is very, very concentrated THC. It's processed with butane, so as I said, it's very concentrated. You put it in a pipe and smoke it. So it's very good to have really good interviewers, very good rapport with your subjects, because we learn all sorts of stuff about how they're doing. Our 25-year-olds, the first maybe 30 that we've done so far, 70% of them are using marijuana. Almost half of those are using daily. Okay.
What would the general population be using?
Repeat the question.
The question was, what's the general population prevalence rate of marijuana use? I don't know. I need to look that up. Thank you. It's not that high. Okay, so I need to thank my co-investigators. This is not something that any one person or any one discipline can do on their own. It takes a variety of expertise. And then, of course, our wonderful staff, half of whom are no longer with us because we, before we started this new phase, people had to go find new jobs. But nevertheless, thank you to everybody, and thank you again for the invitation. Questions?
Well, I can talk loud enough, I think.
They're streaming it.
I'm curious as to, we're having a major opium heroin epidemic and neonates. Is that happening also in Pittsburgh?
Yes, it is. It is. We're starting to explore being able to look at that. There's an initiative in the Graduate School of Public Health looking not specifically to pregnancy, but looking at some of the factors with that. And I have a colleague at McGee in OB who's looking more clinically in terms of treatment of the women. There's a new clinic that's opened up at McGee to administer buprenorphine, because there's the methadone clinics that are separate, and then McGee now has a buprenorphine clinic, partly in response, I think, because private practitioners can prescribe buprenorphine, so McGee wanted more control over their being able to prescribe it to their pregnant population. Okay. We're talking about doing some comparison of prenatal buprenorphine versus methadone, but you think this is hard. The two populations are very different to begin with.
So I'm very interested in the interaction between those drug exposure and also violence exposure. And your mother kind of treat that violence exposure as a mediator. Mm-hmm. And I know that other people also do that in cocaine studying researchers. But I always have some problems with thinking about how that violence exposure can be mediator, because I think it's more like another confounder, but it occurs simply occurring later than prenatal cocaine exposure. Kind of just treating as a mediator doesn't quite persuade me to treat them mediator. For me, it's more like it's very conceptually very related in a way that because of due to lower income population, but it's not necessarily mediator. And we know that violence exposure is a very significant predictor for substance use and risky sexual behavior. So I'd like to kind of hear what your rationale kind of treat that violence exposure as a mediator.
Are you asking because the timing that you think it precedes this?
The reason I think that why violence exposure may not be mediator is because violence exposure and cocaine exposure can be a function of other bigger construct, which is low SCS.
A third variable.
Yes.
That's possible. What I thought, well, let me answer what you didn't ask. This is the child's lifetime exposure, because I know we also have the mother's exposure, in which case, the timing would be an issue because we wouldn't be able to put it in the causal pathway. But because this occurred after this, then, that's the rationale for having it in that timing causal manner. But you're right. You're right. A woman who's using drugs during pregnancy, her kids are going to be more likely to be exposed to violence. We control as best we can other factors that are associated with both of those things. We could do the model differently and treat it as a confounder instead of a mediator, to answer that question.
But that would make a difference in the results, I don't think.
No, I don't think so. Sonia, go to the microphone.
I'm thinking about our co-investigators and also our doctoral students who are very interested in factors that help make, in this kind of scenario with this number of high-risk children, resilient, or things that are assets, and whether or not you've looked at some of those aspects. I also have about 50 other questions, but I'll ask you later.
No, we've been focused on the bad things. But it's a very good point. Looking at things like having a supportive grandmother is very important. Education, pursuing education. So I'm working on a 21-year paper now. I'll have to look at those kind of things.
Wonderful talk. What about the children of these 25-year-olds? Do you see any pattern in their behavior? I mean, that would be a complex thing to sort out because of the environment.
Yeah. As I said, at 21, 30% of the females had a child, so by 25, we expect that probably half will have children. We've always talked about doing a C2. The problem is, in the scheme of epidemiology, our sample is not that big. 200, it's big to a psychologist, but it's not that big to an epidemiologist. And so you figure maybe 100 of the offspring's offspring. And they are such a wide age range that we've struggled with whether we'd be able to get some kind of consistent measures. So we haven't looked at them. But it would be very interesting to do so. Go ahead.
I'm going to jump in. I want to congratulate you on just a remarkable study, and then to carry it out over this long period of time. I know there's resiliency with you and your research.
I'm going to talk about that tomorrow.
Yeah, okay. And also, I was struck by how clever you were in getting interviewers who really could match up and with the people you're working with. Could you just talk about where you recruited them and kept them and trained them?
Okay.
And the second question is, it was surprising to find, if I understood it correctly, that individuals who tested negative in their urine sample but did admit using cocaine, it surprised me that you relied on self-report over the biological measure.
We got more information by self-report.
Yeah, well, so is it that the urine testing, is that unreliable?
The window of detection for a urine screen with cocaine is only 2 or 3 days. So the time frames are different. And because we wanted a broader time frame, we wanted the pattern of use during pregnancy, and we asked the woman, okay, during first trimester, and we went into a lot more detail than that, but just for general. So first trimester, what was her use like? So that's why one reason we're getting more report on the interview than on the urine screen. Okay, that answer that? Okay. All right, interviewers. During our prenatal phases, my memory. I assume we recruit now. The Human Resources Department was a lot different in the Med Center than it is now. Probably didn't even exist back then. We recruited women that we not matched in the statistical sense of the word, but we had women that were both African American and Caucasian to correspond with our sample. They were high school educated. Some of them were starting to take college classes. They grew up in some of the same neighborhoods that the women did. I can't remember the details of where we found them, where we recruited them from. But it was a fine line between a good employee and being able to be comfortable asking the women about their drug use. We had some women that were very knowledgeable about drug use, but they were not good employees. But the ones that were successful, they were very familiar. They were. I won't say they were recovering, because they weren't, but as in they were not addicted. But they were very familiar with the street terms for drug use and made the women very comfortable in talking about it. In our later phases, our interviewers are more typical research assistants, have psychology backgrounds, public health backgrounds, and we do have had to do more training in terms of what the current drug slang is. But they all have very good interviewing skills to put the people at ease in talking about illegal drug use. The other thing that we do is we have obviously the consent form. We tell them we're not associated with your medical care at all, and this was very important in the prenatal phases, that we are not associated with your medical team. Okay? And I had doctors call me during the prenatal phases. This baby's in the NICU, and something's wrong with the baby. You've got to tell me what this woman used. And I said, no, I don't have to tell you what this woman used. Did you try asking her? Well, no, she's not going to tell me. I'm like, well, I can't tell you. But we also have a certificate of confidentiality from the federal government that we explain to the participants that their information cannot be subpoenaed. Not so much a problem in Pennsylvania, some other states. I know our colleagues in Florida were very, very concerned about some of the district attorneys and how they were trying to prosecute women for drug use during pregnancy, which, as you know, is not an effective means of getting women into treatment.
This will be our last question.
Oh, thank you. Lucky me. Thanks so much for your talk. I actually am interested sort of in the last point, partly because over the years, you have so much wisdom in the methodology, thinking about the opiate epidemic here in Ohio and across the country, and looking at the next generation of the researchers. I know we have some locally who are beginning to look at pregnant moms and what are the outcomes for the children. I guess my question is not only what can they learn from you, because I think that's much of what your talk was about, but in particular, you mentioned that much of what you focused on in your research was on the negatives, and I'm curious about what you think the impact of your research has been on stigma, because we know that stigma is such a big factor in people's ability to recover, to seek treatment, to seek prenatal care. And I think moving forward, maybe some of your wisdom on the impact on stigma might make a big difference.
It's a very interesting question. I don't know how you would evaluate that. There have been some editorials addressing that issue. Debbie Frank in Boston has been very active in that. She's very interested in this piece here, this exposure to violence. In her sample, this overwhelms most other effects because of the nature of her sample. I think what I would like to think is that we've helped put things in perspective, that it's not the dire consequences that were originally predicted, not being able to teach these kids. There are not, the huge physical malformations that some of the early case studies were predicting. But that there are subtle effects after you take into account all of these other things, or some of these other things that impact child outcome, child development. Okay.
I want to thank you so much. Can we thank Dr.
Thank you.
Richardson?
Thank you. I really appreciate it. Thank you.
And some of you may have heard we're having a 100th birthday celebration, and so these
Well,
are
thank
some
you.
gifts for you.
Thank you. Very nice.
Birthday gifts. It's a party favor. And if you'd like to continue this conversation, we're going into room 114 right next door for more informal time. And I know some of you are coming to that. Thank you so much.
Thank you. Thank you. I really appreciate it.